All right, students, we are back with another exciting chapter, this time chapter 12. We're going to be looking more at the structure of DNA and how proteins are made in this chapter. So we'll be looking at genetic material, replication of DNA, gene expression, RNA genetic code, transcription, and translation. So this is the process of protein synthesis and how proteins are made. Now, it may sound a little bit confusing, but it's really not that bad, especially when you compare it to some of the other more complicated things we've learned, such as photosynthesis and cellular respiration. This is really kind of a logical process. And this was figured out by a couple of different people. First of all, we had Frederick Griffith. He investigated what is called virulence of Streptococcus pneumonia. So he was looking at pneumonia and the virus of it and how it can be passed from living to from a cell into a different cell and how it transforms. There was further research by this person named Avery and other researchers that worked with Avery that discovered that DNA is the transferring substance. So before this, they kind of had an idea that something was being passed on, but they didn't know what it was. So what is it exactly that is being passed on from one generation to the next? And they eventually were able to figure out that yes, it is DNA that is passing on this genetic information. And they discovered that DNA is the transferring substance, and DNA from dead cells is incorporated into the genome of living cells when it was looked at in this virus formation. So DNA and proteins were the candidate for the hereditary material. They couldn't decide, is it DNA or is it proteins that get passed on? Proteins contain 20 amino acids that can be sequenced and arranged in different ways. So what's interesting about these 20 amino acids is they have not changed very much at all over time. And we have the same 20 amino acids as plants do and other animals do. This same 20 have been around for a while, and they get combined in different ways to make these proteins. So it's considered highly conservative because those 20 amino acids determine many of the proteins, and they determine all the proteins that we have. DNA and RNA each contain only 4 types of nucleotides. And so that's why they are trying to figure out, well, was it the proteins that get passed on or was it the DNA that gets passed on? What they determined, though, is that in order for genetic material to be efficient, it must be able to store genetic information, it must be stable and be replicated because we need to make copies of this information through the mitosis process and meiosis process, and must be able to undergo mutations to provide variability. So whatever the genetic material is, it has to meet those different qualifications. And researchers were able to show that DNA can fulfill all of those functions and proteins not so much. So Griffith injected mice with 2 strains of this pneumonia, an encapsulated S strain and one that did not have a capsule in the R strain. The S strain means that the S strain was virulent, which means the mice died. It has a mucose capsule and forms shiny colonies. So with the S strain, the mice died. With the R strain, the mice lived, and it has no capsule. The reason this is important is if we injected the mice with the capsule, they died. If we injected them without the capsule, they lived. But what if we killed the ones inside the capsule by heating them up? Okay? That should mean that the mice lives because that's what happened. So we killed this virus, put it in the mouse still lived. But now we see something interesting. We killed this one, which should mean the mouse lives. We injected this one without the capsule, which also should have meant the mouse lived. But we find out that the mouse actually died. What was happening was that the DNA was taking over the dead cells, and it was somehow crossing between the two, which was making it active so that the mouse actually ended up dying again. Interesting results considering, in this case, alive, in this case alive, but you combine them together and it ends up with the mouse being dead. So scientists argue that DNA lacked the variability to be able to store the genetic information. Avery and his colleagues used enzymes that break down DNA or proteins in separate experiments to figure out what was actually going on. The only enzyme that had effect was the DNase, which prevented the transformation from occurring. These experiments showed that DNA was the transforming substance. So Avery kind of took the final step that Griffith started to figure out that yes, it was actually DNA. So in his case, Hershey and Chase did another experiment, and theirs was really quite interesting as well. That just kind of sealed the deal as far as DNA being the main genetic information process. They used a virus called T-phage composed of radioactive labeled DNA. Radioactive DNA and the proteins, they could pick. Do we want the DNA to show up as radioactive, or do we want the protein to show up as radioactive? And if it shows up, then that means it got passed on. So they were infecting bacteria with it. What they discovered was that in the case of DNA, it was exhibiting them to be transformed, and the DNA was showing up that yes, it did get passed on. The proteins were not getting passed on, and the protein was radioactive but not being passed on to the bacteria. So here we can see what happened. We had the they allowed it to infect the bacteria, and then they shook it up in a blender to release this capsule, and then they were able to look at what was passed on. In this case, they were able to determine, well, inside of this is the radioactive material. That's what was infected, and that was the DNA. These radioactive capsules, they were released, and they are not being affected. So the capsules were triggered, but they were not what the information that was passed on. The DNA was monitored in this case, and it was the information that was passed on. So Hershey and Chase's experiment was able to determine that yes, in fact, it is DNA that allows the information to be passed from one generation to the other, and not the protein itself. Now, the structure of DNA, as we learned from nucleic acid back when we learned about those macromolecules, it has a ribose, a sugar, and a nitrogen base. A phosphate, a sugar, and a nitrogen base. Phosphate sugars on the side and the nitrogen base on the inside. So the nitrogen bases as the adenine, guanine, thymine, and cytosine. The adenine and the guanine are called purines, and the thymines and the cytosines are called pyrimidines. The purine and the pyrimidine can only match up with each other. So adenine and thymine always pair, and guanine and cytosine always pair. Because of this pairing, we only need to know one half of the structure, and we can predict what the other half is going to be because they always pair up the same way. So the amounts of A, T, G, and C in DNA are consistent among the members of the same species. They vary from species to species. One thing they noticed is that for every adenine, there is the same amount of thymine. For every guanine, there's the same amount of cytosine, and vice versa. So this suggested that A and T always paired up, and G and C always paired up. You can remember it by @GC. All of this suggests that DNA uses complementary base pairing to store genetic information. Each human chromosome contains an average of about 140 million base pairs. So it's a very big sequence that has lots of different combinations. So here we can see our nucleotide bases. We've got our phosphate and our sugar and our nitrogen base. Phosphate, sugar, and nitrogen base. Adenine, guanine, thymine, and cytosine. And in humans, you can see the different comparisons here compared to fruit flies, corn, fungus, and 2 types of bacterium. So the numbers vary based on which species they are in. So then there's this whole Rosalind Franklin thing. Rosalind Franklin was able to make a picture of DNA using X-rays, and she found that if it was concentrated, she was able to get a diffraction photo, an X-ray diffraction photograph of DNA. And this photo was vital for the development and the identification of the double helix structure. A colleague of Franklin's, Wilkins, showed her photo to James Watson, who understood its significance about the DNA structure. So Rosalind Franklin was the one that took this X-ray diffraction photograph. And Watson and Crick are the two that often get credit for discovering the double helix shape. Here's what the X-ray diffraction looked like, and it's very difficult to make sense of, but this picture was, and you can see here, X-ray beam with a crystallized DNA, and then it made this diffracted image. This gave rise to that double helix pattern, and it was because of this picture that was taken by Rosalind Franklin that allowed Watson and Crick to make their model. So the double helix model is similar to a twisted ladder. I'm sure we've all seen it before. And it has the sugar and phosphate backbones, and then the rungs in the center of the ladder are made up of those nitrogen bases. The nitrogen bases are joined together in the very center by hydrogen bonds, which are vital for the separation of the two strands of the two sides of the DNA. This allows them to break apart in the middle and come back together, and it also holds it in that spiral shape. This complementary base pairing of AT-GC, AT and GC, it allows them to make a copy of the DNA molecule, which we'll see with the replication process. Watson and Crick did receive the Nobel Prize in 1962 for their supposed discovery of the DNA double helix, but Rosalind Franklin may have been the one to initially discover it because it was her picture that led to that final discovery. The other important thing is that DNA is read from what's called the 5' to the 3' direction. So one strand, it's like one strand is going one way and the other one is going the opposite way. So they kind of lay like this. One going one direction, one going the other direction, and one of them's like flipped upside down. So it's going 5' to 3' laying in opposite directions from each other. So here we can see what the basic model looks like. We've got our double helix, we've got our sugar phosphate here on the side, and our nitrogen bases in the center. We've got our thymine and our adenine and our cytosine and our guanine, and it's the way that these get matched up that actually determines our genetic code and our protein sequence. These hydrogen bonds here in the center can constantly be broken and reattached, allowing for us to read that genetic code. This is a picture of Watson and Crick with their double helix model that they supposedly created. All right, so DNA replication, the process of making a copy of the DNA, which is vital for the mitosis process. In order for mitosis to occur, we want an exact copy of the DNA. We want it to be exactly the same as the original parent, and that's what replication is, making a copy of the DNA. This is called a semi-conservative process because we only need 1/2 of the DNA in order to make a template and make a copy of it. So basically what we do is we split the DNA down the center, and then we use each strand or half of the ladder to use as a template to code for a new strand. So 1/2 is original and 1/2 is built new. And that's why it's considered semi-conservative. So here we can see this process. Here's our original strand, the parent. We break it in two, and so 1/2 of the ladder is built upon, and 1/2 is old, and 1/2 is new. And because of the complementary base pairing, T always pairs with A, G always pairs with C, we can guess what's coming next. We know that, okay, this one's going to pair with A, this one's going to pair with C, this is an A, G, and so on, because it's always going to have the same pairing. So we only need 1/2 of this ladder in order to make an exact copy. So that's what happens. The hydrogen bonds break in the center, and then we have an exact copy made. Semi-conservative because 1/2 is an old strand from the original, and 1/2, the green, is a new strand that was constructed by the cell. Now, how this works is you have an enzyme called DNA helicase. DNA helicase is going to unwind or separate the two strands and make them into single strands, or half of the latter. The single strands binding proteins attach to newly separated DNA to prevent the helix from going back together or to keep them separate. Then we have the complementary base pairing, which is the at GC again. And there's something called DNA primase, which places short primers on the strands to be replicated, saying we need to replicate from this place on the DNA to this place. And so then we have something called polymerase. Remember, anything that ends in ASE is a type of enzyme. So DNA helicase is an enzyme. Polymerase is also an enzyme. It recognizes RNA and begins the DNA synthesis process. There are leading strands and lagging strands because remember, it can only be read from the 5' to the 3' direction. So we have to go 5 to 3. We can't go 3 to 5. So the leading strand is going 5 to 3. The lagging strand is going 3 to 5. And the lagging strand is a little bit weird. It has to be built in chunks. It can't be built in one straight continuous piece. It has to be built in a chunk and then a chunk and another chunk because it's the lagging strand. Each daughter DNA molecule contains one old and one new strand. Now, this would be another great process that you should watch some YouTube videos about because it's really helpful to see this as an animation to see how the lagging strand and the leading strand are built. So watching a YouTube video on this would be really helpful, and there's many good YouTube videos out there. Here we can see how the process is working. We've got our parental DNA, and we've got the DNA helicase that is coming through and separating those hydrogen bonds. We have the SSB, which is saying, okay, we're going to start here, and this is where this process is going to start. DNA polymerase is coming in and matching up those materials. Because we have a leading strand and we can go 5' to 3', it's just chugging along, ripping out that DNA, building new molecules using a template strand and then making a new strand, but it can continually build it. Our lagging strand, however, does not have such luck. We have to build it in chunks because we're reading the opposite direction. So we have to build it in a chunk here, to build it in a chunk here. These chunks are called Okazaki fragments because Okazaki, somebody discovered these fragments. When you discover something in science, you get to name it after yourself. So the Okazaki fragments are added in these chunks because, again, this is the lagging strand and it cannot read in a continuous pattern because it's kind of reading backwards and it can only be read in the 5' to the 3' direction. Again, this would be a really good thing to go into YouTube and watch a video about. The animation would help it make a lot more sense. Now, in some prokaryotic things, such as bacteria, they have a single circular loop of DNA, and replication moves around the circular DNA in both directions. It produces two identical circles, and the process begins at the origin of replication, and replication takes about 40 minutes, but the cell divides every 20 minutes. A new round of replication can begin before the previous round is even completed because it's going in a circle. So that's kind of neat to imagine that replication is taking place faster than the cell can even divide. DNA is the replication begins at numerous points along each linear chromosome, and DNA unwinds and it unzips into two strands. Each old strand of DNA serves as a template for a new strand, and with the complementary base pairs, we can usually figure out what's going where. Requires enzyme DNA polymerase for this to take place. This is another really good summary slide that you may want to screenshot and come back to because it does explain the process really nicely. So replication, the bubbles spread bidirectionally until they meet. The complementary nucleotides are joined to form new strands, and replication, again, is semi-conservative. This means that there was an old strand and then a new strand that was built. Polymerase is unable to replicate the ends of the chromosomes, called telomeres, and telomeres are copies of the enzyme telomerase. There is some speculation that telomerase is one of the reasons that we are aging because as the cell makes copies of itself, telomerase is going to shorten over time, and eventually those telomeres become so short that replication is no longer possible and the individual is going to pass away. Unregulated telomerase activity negatively affects cell function. So if those telomeres gets too short, then that could cause disruption of the cell replication process. So here's prokaryotic versus eukaryotic. You can see that in a eukaryotic, it's in a loop, and the process, it can continuously make a loop, whereas if it's in a strand, you can make replications in different parts of the strand at the same time, like these replication bubbles, so to speak, or you can replicate the whole thing, depending on what you're replicating and what your needs are. DNA polymerase is very accurate, yet makes a mistake about once every 100,000 base pairs. It's capable of identifying and correcting these errors, which is pretty important because if you're making a copy of DNA, you really don't want a mistake to happen. If DNA has mistakes, this is where mutations happen. Once mismatched nucleotide is removed, it changes direction and resumes making the DNA. So it can correct itself. Our body has in place mechanisms that allow for correction of genetic mistakes. Hopefully they work, and most of the time they do. Now let's talk a little bit about RNA. RNA comes into play when we are wanting to make proteins. Replication, exact copy of the DNA, is really just helpful for reproduction and when the cell needs to make a copy of itself. But let's say the cell doesn't want to make a copy of itself. It just wants to make a protein, and that protein information is hidden on that DNA strand. The DNA strand does not need to make a complete copy. It just needs to make a little bit. And so that's where RNA comes in. Archibald Gerard described inborn errors of metabolism. Beadle and Tatum experiments on the red bread mold fungus and proposed that each gene specifies the synthesis of one enzyme. One gene, one enzyme hypothesis. So they were able to determine the flow of genetic information is from DNA to RNA to protein to an observed trait. All of our protein information is hidden on our DNA strand. We need to get that DNA information off of the DNA strand and then transfer it to making an actual protein. And that's where the process of transcription and translation comes in. So the mechanism of gene expression, DNA in genes specify information, but information is not structure and function. We've got to figure out how to get that information from the DNA and into a protein. So RNA is what does this. RNA is single-stranded. RNA can leave the nucleus. RNA is more stable than DNA outside the nucleus, and RNA can basically carry information from the genetic code to make a protein. So RNA has several functions. There's several different kinds of RNA, but RNA controls the primary structure of a protein by reading the information from a DNA, taking it out of the nucleus, and using the code that was taken from that DNA, transcribed to an RNA, and then is translated into a protein. So here's how the flow of genetic information works. We have our DNA. We want to get this information out of the cell. So then we transcribe that information to RNA. That's the transcription process. One of the things you'll notice is that there is no thymine on an RNA strand. Thymine is replaced by uracil. Uracil instead codes to the A, or matches to the adenine. So adenine and uracil match up. Guanine and cytosine match up on the RNA strand. Transcription is taking the information from DNA and transcribing it to RNA. mRNA, M for messenger RNA. Messenger RNA takes this information out of the nucleus. We can also see something called a codon here. A codon is a group of 3 mRNA strands. So here, not strands, mRNA groups. 1, 2, 3, that's codon 1. 1, 2, 3, that's codon 2. 1, 2, 3, that's codon 3. These codons, looking at them in groups of 3, are translated into amino acids. So a group of 3 represents 1 amino acid. So in this case, serine. This group of 3, GAC, is translated into aspartate. And this group of 3, CCC, is translated into proline. So these are all amino acids that get strung together based on the RNA, which originally came from that DNA code. Again, reading from the 5' to 3' direction, and here you can see the other 5' to 3' this way. First process is transcription to an mRNA strand. mRNA strand leaves the nucleus and is translated at the ribosome to the polypeptide or the protein strand with a string of amino acids. So RNA is a polymer. It's a type of nucleotide, and RNA contains the sugar ribose instead of deoxyribose. It has uracil instead of thymine, and adenine and cytosine and guanine. Uracil replaces thymine, and thymine we only see in DNA. There's messenger RNA or mRNA. There's also something called tRNA, which is transfer RNA. And there's rRNA, which is ribosomal RNA. Ribosomal RNA is basically a ribosome. Transfer RNA is going to bring information to the mRNA, which is the messenger RNA, and the messenger RNA gets the code from the DNA itself. So here's how the process works. Here we can see that we have an RNA strand. We know it's an RNA strand because, first of all, it's one-stranded, not double-stranded, and it has uracil instead. We got this from our genetic code. So our DNA strand was here, and our DNA had a C, it had an A, it had a T, and it had a G. So we have transcribed it to an RNA, and now we need to translate it to an amino acid and to a protein, reading from the 5' to the 3' direction. So RNA, comparing and contrasting differences between RNA and DNA, we have ribose and deoxyribose. Base pairs, the only difference here is uracil, and in DNA we have thymine. Single-stranded versus double-stranded, and no helix, but DNA does have a double helix. So now we've got our information off the DNA onto the mRNA strand. The mRNA strand has left the nucleus and come out into the cytoplasm. Well, now we got to get that information off of the mRNA strand and turn it into an actual protein. And that's what the codons do with the tRNA. It all goes to the ribosome. At the ribosome, it's going to start to read that mRNA pattern. And that is going to start telling us what the codons are. There are 20 amino acids, and the codons, those groups of 3, the 3 nucleotides, code for the 20 amino acids. So there are codon charts that help with this. There are 64 possible codons, 64 possible combinations. The codons can be redundant, which means that they can repeat. Sometimes there's more than one codon. Excuse me, throat tickle. Sometimes there are more than one codon that codes for the same amino acid. For instance, both UGU and UGC are coding for the same amino acid cysteine here. That means that if there was some kind of mutation and we were supposed to have UGU, but something happened and we got UGC instead, ultimately it's not going to matter because it makes the same amino acid. This also allows for some mutations to take place if it's not caught previously, because we have what's called a silent mutation. We've got UGU, which mutated to UGC, but they code for the same thing, so it doesn't matter that there is a mutation there. Every codon only codes for one amino acid. Here is our basic codon chart, and here's how to read it. Let's say we had UGU right here that we saw. Okay, so U, our first base is U, and then our second one was G, so UGU. So UGU is right here. So our first one, our second one, and our third one, UGU. UGU codes for cysteine. Find it on the chart, and that's our amino acid. But it's not the only one that's cysteine. Here's another cysteine. These stop ones become important later on because these stop ones are like a gap in the amino acid chain. It's like when you're building that necklace and you're stringing all these beads onto that necklace, eventually you need to cut the necklace off because it's long enough and you're done. That's what these stop ones do. They make a break in the chain, and that means that the protein is finished. Most of the time, pretty much all the time, you're going to start with AUG, which is the methionine, and that's your start codon. So you start with this one and you end with one of those three. This is universal, which is really interesting. It means that with very few exceptions, all organisms use the same code the same way. All of these are the same. Plant, animal, whatever. If it's alive, it's made of these same amino acids, which is really interesting and also serves as evidence that we all came from one common ancestor, even plants and animals, because they all came from one original organism. We all have the same amino acids. They code for the same 21s, and the same 64 codons code for the same 20 amino acids. The universal nature of the genetic code provides strong evidence that all living organisms share a common evolutionary heritage. Because the same genetic code is used by all living organisms, it is possible to transfer genes from one organism to another. Interesting with gene therapy, that you can get gene therapy from animals because the genes are the same, whether it's in an animal or a human. There's only one start signal, but there are three stop signals. Just like I mentioned, one start signal is AUG, and the three stop signals are mentioned here. So a single chromosome is a very long double-stranded DNA, and that contain hundreds or thousands of different genes. While we may not need all of those genes, we can just break apart part of the DNA that we need in order to transcribe it. So the information within a single gene directs the synthesis, remember synthesis means to create, of an RNA molecule. For genes that code for proteins, the resulting mRNA sequence determines the sequence of amino acids. We get our information that we need from to create a protein from the DNA strand. DNA strands can contain many different portions that code for different proteins. So during transcription, a gene gets unzipped to expose the A, the T, the G, the nucleotides. And then the mRNA comes in and reads that DNA. RNA polymerase is used instead of DNA polymerase. We had DNA polymerase during the replication process. But for transcription and translation, we use RNA polymerase because we're not building another DNA strand. We're building an RNA strand. So RNA polymerase is going to come in, and this occurs in three processes called initiation, elongation, and termination. Initiation is when RNA attaches to a region and it has a DNA promoter, and this starts the transcription process. Elongation is the RNA polymerase moving along from the 3' to the 5' direction and attaches those RNA bases. And then termination is when it gets to the stopping point of the DNA sequence, and so the mRNA strand is able to break off, and now called an mRNA transcript. Here's how that works. We've got our RNA polymerase that is building. We've broken off the DNA that we need. We have these free-floating pieces of nucleotides in here that we're matching up to build our mRNA, and then it's going to rejoin the DNA double helix back here once that part has been read like it needs to. The pre-mRNA is modified or processed before leaving the nucleus to make sure it's stable to go out of the nucleus. There's usually a cap on the 5' end, and that cap is a modified guanine nucleotide. And there's also something called a poly-A tail. The poly-A tail is 150 to 200 adenines on the 3' end. Now this poly-A tail helps to stabilize the mRNA when it leaves the nucleus. The longer the poly-A tail, the longer that the mRNA can stay stable outside of the nucleus itself. So long poly-A tails are usually for more complicated RNA strands to help them remain more stable. Pre-mRNA is composed of exons and introns. This is kind of interesting as well. The exons will be expressed. The introns occur in between the exons. So it's kind of confusing. The exons stay and the introns leave. So the exons are expressed and the introns are in the way. It allows a cell to pick and choose which exons go into a particular mRNA. The exons stay, the introns are taken away and removed. So basically, before it is even translated into an amino acid, we're cutting chunks out. That's what's happening. Those introns are being cut out and the exons are being mushed together. So think of like a long strand and you're cutting out pieces and mushing the pieces together that you want to keep. And this is RNA splicing. So we're splicing the segments that will not be expressed are the introns, they get removed, and the segments that will be expressed are the exons and they stay. So we're removing bits and pieces and pushing together the parts we want. In low eukaryotes, the introns are removed by self-splicing. In higher eukaryotes, the removal is done by spliceosomes that contain small nuclear RNAs. The spliceosomes use a ribosome that are going to kind of cut them out and cut the pieces out and then mush the rest of them together, so to speak. So here we can see kind of the process. We have our DNA, but we only want the exons, and so the introns are going to be removed. We've got our cap and our poly A tail, and then we are able to splice those introns out until we just have the exons present with our poly A tail. Now, once we've removed those pieces that we don't want anymore, now our mRNA can actually exit the nuclear membrane. So as complexity increases, the number of proton coding genes does not keep pace. The proportion of the genome that is introns increases. So introns, why do we even need them? So exons might combine in various combinations, would allow different mRNAs to result from one segment of DNA. Introns might regulate gene expression, and introns may encourage crossing over during meiosis, because if there's a gap of introns that we don't necessarily need for coding information, well, let's just cut off that piece and switch it with another homologous chromosome. So an exciting new picture of the genome is emerging the more we understand of introns and exons. So that is our first step of transcription. Now we've transcribed the information. Now we need the second step, which is translation. During translation, the mRNA codon sequence is translated into a protein or amino acid sequence. So now we've got the information from the DNA. We put a poly A tail on to make it stable. We've gotten rid of those introns, which we don't really need. So now we've got what we need. It's exited the nuclear membrane. Well, let's figure out how to get that information to an actual protein. So it's going to join up with an rRNA, which is a ribosome. And the ribosome is going to read this mRNA that we've created. The mRNA has 3 binding sites for tRNA. tRNA is transfer RNA. Its job is to bring the amino acids to the ribosome, match up with mRNA, and make sure that the right amino acid gets to the right spot in the right order. So the tRNA brings the amino acids and basically matches up to the codons of the mRNA. So the tRNA has an anticodon, which matches to the codon. And there'll be a picture coming up that kind of makes sense. So just kind of bear with it for right now. The tRNA has the amino acid attached to it. The tRNA has an anticodon that matches to the RNA, the mRNA. There are approximately 40 different types of tRNA are found in most cells, and there are fewer tRNAs than codons because some tRNAs can pair with more than one codon. Wobble hypothesis, that's a little bit strange of a name, predicts that the third position in the tRNA codon doesn't obey the AUGC configuration rule. And some tRNA molecules can recognize as many as 4 separate codons. So all that to say, the tRNAs can be used sometimes for multiple amino acids. Now the tRNA kind of looks like a T, I think, even though it stands for transfer RNA. Looks kind of like a T. Here is our anticodon. Remember, a codon is a group of 3 nucleotide sequence that's found on our mRNA. Here's our mRNA strand. Here's our anticodon, which matches up to that, and that is going to bring in an amino acid. So here is our little bead of amino acid that is coding for this particular codon. So we've got that matching up, and our tRNA has brought it in. And all of this is taking place in a bigger unit that is the ribosome. Again, this would be an excellent time to watch a YouTube video about the transcription and translation process, because if you see this in motion, it would really help make it a lot more sense. But here is our anticodon, and here is our amino acid. It looks kind of funny when you're looking at it in a 3D pattern. So the ribosomal RNA produced from a DNA molecule in the nucleotide, a completed ribosome has 3 binding sites to facilitate the pairing. So again, codons, 3 codons, and we have an E for the exit site, a P for the peptide site, and an A for amino acid site. The larger ribosomal subunit has enzyme activity that creates the bond. It's a peptide bond, and so is why it's called a polypeptide that joins the amino acids together. Think of beads on a string again. The beads are the amino acids, the string is the peptide bonds. The peptide bond is created many times to produce a polypeptide, and this polypeptide will fold over on itself and coil around itself to make a 3-dimensional shape. So here we have our ribosome. Our ribosome actually has 2 units, a large subunit and a smaller subunit that join together and kind of wrap around our mRNA. Our mRNA goes inside, and we have our codon that's in there. As the RNA or the ribosome moves down the mRNA, it exposes more and more of the codons to the ribosome, and the ribosome is reading 3 at a time. That 3 is going to get matched up to a tRNA. The tRNA drops off its amino acid, the ribosome moves further down, and then the empty tRNA is able to leave, and a new tRNA is able to come in. And you can see here how it's building up those amino acids over time. Now, the components necessary for initiation are, in order for this translation process to actually occur, you have initiation, which is the small ribosomal subunit, an mRNA, an initiator tRNA, large ribosomal subunit, and initiation factor. All those things have to come into play before the protein can be made. All of those things have to meet up in order for this process to happen. The initiator tRNA always has the anticodon UAC. That methionine we looked at on that chart. That is our initiator codon. It tells us, okay, let's start here. It's capable of binding to the P site within the mRNA. So we have that start codon, which is AUG, and then the initiator, which is UAC, because that matches up. UAC matches up with AUG and attaches to the P site of the large ribosomal subunit. So here we can see it sliding along our mRNA strand. We have our small subunit, our larger one, our ribosome. We have our codon, here's a codon, and our tRNA and our mRNA. And then we have our first amino acid, which is always met. And then elongation is the next step. So that's the ribosome is moving along the mRNA, adding more and more amino acids as it goes, and the amino acids are getting joined together by those peptide bonds. Then the second tRNA moves on, so this is called translocation. So once the amino acid is joined, that tRNA that has dropped off its amino acid can leave, and the ribosome moves on to the next codon so that another tRNA can come. So again, it's just kind of a creep along the mRNA strand. So this one has already dropped off its amino acid, and so it can go away. And that means that the ribosome moves down the mRNA strand, exposing another codon, which brings in another tRNA. Then the ASP is going to be joined to the VAL. This one is going to drop off its amino acid because it gets joined onto here, and that means that this tRNA is able to move away. So here we can see our anticodon coming in. The VAL is going to get transferred over here. This codon is now empty, and so that codon can go away. The ribosome is going to scooch over one, exposing a new one, and so on. So this one is going to get joined onto the ASP. then this tRNA is going to move off, and this one is going to be attached, and then the ribosome is going to move on. So it's kind of a creeping factor. Again, looking at a YouTube video is going to be very helpful for this. tRNA moves to the P site and does not code for an amino acid. A protein is going to be determined by the termination process. Termination is when one of these three is met, and that leaves a gap in the site, and that gap is going to prevent it's like an opening or an empty space. The ribosome releases from the mRNA and disassociates. So a termination process, we have that gap protein or that gap that appears, and here is the entire process with it being transcription, and then being modified, coming out into the cytoplasm for translation. We have the tRNAs coming into the ribosomes, being read by the mRNAs, and this would be another good slide to just kind of pause and study for a little bit to make sure you understand the process. Transcription taking place in the cell or in the nucleus, and then translation all taking place in the cytoplasm with your polypeptide chain being formed. All right, so over time, all of this is going to join together to make a larger protein. And some ribosomes are free in the cytoplasm, some are attached to the rough ER, and they are modified using that same highly conservative code and the codon chart coding for those amino acids. So we've made it. That is the end of this chapter, and we'll see you guys for the next one. Bye.